Our project is titled Investigating Genes Specific to Fighting P. aeruginosa Bacterial Infections in Cystic Fibrosis. Cystic fibrosis, or CF for short, is considered an inherited genetic disease that causes patients with these conditions to suffer from increased bacterial infections in their lungs. Buildup of mucus in these bacterial infections can make it extremely difficult to breathe. Thick mucus is the perfect environment to allow bacteria to grow, and is why so many CF patients struggle with bacterial infections. Current treatments for these infections include antibiotics. In theory, they're a great option because they can kill a wide variety of bacteria. However, they often lead to antibiotic resistance, making the bacterial infection increasingly difficult to treat. Bacteriophages offer an alternative treatment option. Bacteriophages are viruses that infect bacteria by injecting their genetic material into bacteria to kill them. They have unique abilities to target and combat specific types of bacteria, unlike antibiotics, which kill a broad range of bacteria, including healthy ones. This also avoids bacteria becoming resistant and running out of treatment options, as there is an estimated 10 bacteriophages for every bacteria type. This specific targeting ability makes bacteriophage an exciting alternative treatment option for CF. The goals of this research was to identify which genes are essential to fighting P. aeruginosa bacterial infections for cystic fibrosis. A CF clinical trial combined 10 phages for the treatment of CF patients. This cocktail of bacteriophages were specific in targeting strains or variations of the bacteria P. aeruginosa. Our team then found the genomes of each of these phages and used a genome comparison map to find genes of similarity in at least 7 of the 10 phages. These similar genes might be key to fighting these types of infections. Our team was able to find and analyze 3 genes of similarity across at least 8 of the phages. For simplicity, we called them genes A, B, and C. Gene A is seen here in the poster in brown. All of the brown-colored genes in this image correspond to the similar genes that were grouped to call gene A. All 10 phages had a variation of gene A, making this gene seem promising as a gene that is specific in targeting P. aeruginosa bacterial infections. Of these genes, many of them were reported to have no known function. However, 3 of these variations of gene A reported functions. They included 5' to 3' exonuclease and putative cell wall-associated hydrolase. These are very different protein functions that seem to not have a relationship between them. However, they all seem to play a role in phage survival and its life cycle. Gene B is seen here on the poster in pink. 8 of the 10 phages have a variation of this gene. Of these 8 genes, almost all of them are hypothetical proteins, and only 2 have function, which is a putative structural protein and DNA polymerase. It should be noted that the function DNA polymerase was also found as a possible function for the sequence of gene B. Given how the function is vital for DNA replication, this could be important for the effectiveness of a bacteriophage. Gene C can be seen here in the poster in teal. 8 of the 10 phages have a variation of this gene. Of these 8 genes, 5 of them called hypothetical proteins, but 3 called putative proteins. 2 call for a putative tail fiber protein and 1 for a putative RNA methyltransferase. Tail fiber proteins play a role in the virus's ability to attach to their host, while RNA methyltransferase could help the way the virus interacts with the host cell. This probably both play a role in the way the virus infects and attaches to the host bacteria cell, so this could be an important gene in these phages. Because these genes are similar in many of the phages found, they might be key genes needed in the phages to fight P. aeruginosa bacterial infections in CF patients. 3 genes were found to be similar across at least 8 of the phages and used to treat cystic fibrosis. No functions seem to be linked to these genes, but this might be determined if more phages are compared in the future. Overall, these similar genes could be key factors in what make a phage prime for fighting these types of infections. Most of these genes were recorded to have no known function, but were very similar in amino acid sequence. More wet lab work in identifying the function of these genes would be helpful in piecing together what makes these phages special in targeting P. aeruginosa strains. By identifying these key genes, it may make it easier to screen newly discovered bacteriophage or even add discovered bacteriophage to see if they would be beneficial for treating CF patients.